Hello, my name is Sein Jeung, and I'm based in Berlin Mobile Brain Body Imaging Lab led by Klaus Gramann, and also affiliated with MPI Leipzig led by Christian Döller. So I'm going to talk about EEG dynamics that's associated with real-life navigation using full body motion, especially in patients in hippocampal lesions. When we are about to talk about navigation, I want everyone to think about how we navigate in everyday life. You first exit the subway station, and then first thing you do is perhaps to look around and identify a landmark that can give you some sense of orientation. Another important strategy is to keep track of how you have walked and maintain the heading relative to where you started from. So I'm going to term these two strategies landmark-based and body-based strategies. And humans use a variety of strategies to navigate or to find our way through space. The point of the whole talk is that sometimes in a highly controlled environments, the use of body-based strategies are limited. So we might be looking at a process that is not only qualitatively, but also not only quantitatively, but also qualitatively different from how we navigate in everyday life. And this is especially true for navigation because it is an intrinsically embodied process. This is a video that I really like from the 90s where virtual agents are simulated with two different modules. So one is the control system, which corresponds to the brain, and the other one is the body, which has restrictions on joint angles or the forces that it can exert. And what happens if you give these virtual agents the fitness function and some rules around the environment, they naturally develop a very nice repertoire of motion like swimming, walking, jumping, and following. And this is probably how real animals have evolved. So in rodent research, we know that the hippocampus in the medial temporal lobe houses cells that are crucial for map-like representations. These include hippocampal place cells and entorhinal grid cells, and these are mainly used for landmark-based or environment-anchored coding of the space. We know that this grid cell-like activity, due to the geometric structure of the grid cell firing pattern, can be read out in the Bol signal using fMRI. But the problem is that if you are observing navigation behavior in scanner, the embodiment aspect becomes questionable. So in this paper, they observed the effect of embodiment on grid-like coding by showing participants laying inside a scanner a virtual avatar of their body. This led to some behavioral differences. As you can see, the distance from the border of the participants was higher when they were shown this avatar of themselves laying in the scanner. And then the distance error was decreased in the body condition compared to no body condition. What's really fascinating is that the grid cell-like representation was only observed in the no body condition. We can speculate that this is because the reference frames being perpendicular to each other, laying inside a scanner and having this illusion of being shoved around on a bed is probably not very naturalistic, and it confuses our brain in some way. So this has been a question that's been around for some time. So what are the alternatives that we have? Maybe we can turn to electrophysiology, but the medial temporal sources are difficult to investigate because they're situated deep inside the brain. So either we can, due to technological advances these days, we can apply EEG in mobile setup. So Martin Sieber, after my talk, will talk a little bit about EEG in mobile setup. And we can also use MEG, but MEG is also highly susceptible to motion artifacts. So maybe when we have very good models about the activity pattern, we can bring this to non-invasive and mobile setup. So what I'm going to present today is a collaborative lesion study so that we can use it as a pointer to electrophysiology features that are associated with behaviors and that are supported by the affected area. So we had 10 patients who had right medial temporal lobe resection. One participant was excluded from EEG analysis due to spectral artifact, but was included in the behavioral data. And we had 2 age and sex matched controls per patient. What they did was to perform the same task in 2 different setups. In one session, they stood in front of a screen and controlled their virtual camera motion using a joystick, just like when we play first-person perspective games. And in another session, they were allowed to walk around freely, untethered, with a VR headset, gaming VR headset, plus 128 channels, high-density mobile EEG, powered by the batteries attached to the backpack. This is the view that was presented to the participants, so relatively cue-rich naturalistic setup. And the test that they performed was this classical study used in rodent research, Morris Watermaze task. So the basic idea of the task is that they walk around this circular arena until an object pops up. And when it does, they remember the location. And then in later trials, they need to navigate back to the hidden object location. And they performed it from 4 different rotated starting points. So this is the result. So as you can see here, the heat map shows the presence probability in probe trials in medial temporal lesion resection group. And in the stationary condition, you can see that this is the target location, the circle here. The presence probability is not particularly condensed around the target area. So this shows that they didn't really form a robust spatial memory of where the target was. In comparison, healthy control participants show more clustering of the presence probability around the target location. This impression changes as soon as we move them to the mobile setup. The clustering around the goal location is more clear in the patient group, and even nicer in control group. Like you can really see that they were precisely locating to the target location. If we compare the memory score based on the Euclidean distance between the target location and the response location, there is also this clear interaction. So both groups benefit from being able to move their body through physical space, and this benefit is more pronounced in the patients who are missing the right medial temporal lobe. Not the entire lobe, but like has a lesion in the right medial temporal lobe, which is the side that is typically more linked to navigation performance. But are they actually using the body-based information that is additionally present in the mobile setup in different ways? The answer is yes, because we looked at the behavioral measures that can tell us something about these strategies. So first measure we looked at was the angular velocity at the onset of each trial. So I will just remind you of this meme of a person looking around to sample the landmark-based information from the environment. And as you can see in the mobile condition, they both rotate their head more, but this is more pronounced in the healthy controls that are presumably using more landmark-based strategy. On the other hand, as an index of body-based strategy, we looked at the dynamic time warp trajectory distance. So if you are using an embodied strategy, such as I went 3 steps forward, turned left, and then 2 steps to the left, then both trials, like one trial and the previous trial from learning session, will be close to one another. So lower dynamic warped trajectory distance is an indicator of this kind of embodied trajectory replication behavior. So, and if we look at this measure, as we move from stationary to mobile setup, the controls don't really show much difference in this measure, but only the patients with lesions show the drop in trajectory distance between trials, meaning that they probably prefer the strategy of replicating their paths and is supported by more proprioception and vestibular input. For the EEG part, we looked at these frontal midline electrodes in the theta band activity because this is an implicated area in navigation literature at decision points or in active landmark processing. In the baseline trials, they didn't really show too much difference between the groups, neither in stationary nor in mobile condition. And all the EEG data that I'm showing is baselined by the activity from the non-navigation walking session where they followed a waypoint without really retrieving or encoding any spatial memory. So during desktop navigation, we see that the theta is higher in patients versus controls. And this was confined to the epochs that we extracted from the middle section of the trial. This impression is different when we look at the mobile condition, because the pattern is reversed. Now the control participants have higher theta, FM theta activity at the start and at the end of the trial. What do we make of this? So when we now correlate the activity of frontal midline theta from the middle section of the trial, it correlates negatively only in patient group. If you think about it, when their preferred strategy is to replicate the trajectory in the stationary condition, they're unable to continuously integrate their paths. And this difference would be more pronounced not at the end of the beginning or the beginning of the trial, but during the continuous integration part, which is in the middle section epochs. On the other hand, in the mobile condition, the theta difference in the start of the trial show positive correlation only in control group. And this is the critical time window for landmark-based strategy as shown in the behavioral measure of the angular velocity. So to summarize, when we just have stationary setup observation, we would have inferred that they don't really form a good spatial representation of the environment. And then we make some inference about the theta features and how this correlates to behavior. And without the full picture, we cannot really interpret this with much clarity. So what I want to emphasize is that like each of these steps is very important. So first, finding out about the basic mechanisms in animal research, and then we generalize it to like human setups in fMRI, for instance, and then eventually we bring it out into the real world and sometimes circle back to the more controlled laboratory setup, because in real world we stumble upon processes that were previously not known to us. With this, I would like to conclude the presentation and thank my co-authors and collaborators, especially teacher Iggena, who collected the behavioral data and EEG data with me and did all the behavioral analysis. The publication is found here in the QR code, and then Berlin Mobile Brain Body Imaging Group, as well as the tech support and the patients who were there during COVID time, and German taxpayers for making this research possible. Yeah, thank you.